This is such a great pleasure for me to be in this event. I really get used to not saying no to Amos Corzine. I tried it several years ago, and that's not going to work well. I wish you, Amos, well, and I hope that there'll be many happy returns to such a meeting. And now I can share my screen. Sorry. Okay. So I will be talking about discovering of resiliency genes to Alzheimer's disease. And the story is such that in human evolution, lifespan has been somewhere between 19 and 35 years for 50,000, 100,000 years of human evolution. And it's really in a very short period of like 100, 150 years that all of a sudden life expectancy went up to what it is now, 78, at least in the United States, less for men, more for women. And this is the deal that we got by expanding lifespan so much. We got age-related diseases that we've never known before. And they accumulate, and we have to treat them. And so we accumulate one disease and another disease and treatment and another treatment and really bad quality of life. And we need to create a new history for this bad deal that we have. And I want to emphasize that part of getting from age 30 to age 80 are preventative measures, such as harnessing agriculture, cleaning the water, sewers, immunization. We also had things like surgery and antibiotics. But the question is, can we have another way of prevention to extend our health span and possibly our longevity? So we know that there is a biology of aging because we basically know who's old and who's young. What people don't realize is that this biology drives the diseases we have. And Alzheimer is in red because, you can be born with an ApoE4 homozygosity. And you're not going to be demented when you're 1 year or 10 year or 20 year. It takes 60 years or 70 years to be demented. So this doesn't happen without aging. And it's the same with other things. So this biology is very important for the appearance of disease. The good news is this biology can be targeted. And we know that we can target the biology and delay aging, in some instances, stop and reverse that. Now, clinically, when we age, we have all those problems that we can complain about, including decline of cognition. But as biologists, what we have is what we call the hallmarks of aging. It's hallmarks that we agree on. And it's hallmarks, and in order to be a hallmark, you have to show that something goes wrong with aging, and that when you fix it, you get an increase in health span and lifespan in animals. The interconnectivity there means that you don't have to treat all those hallmarks to get an effect. You treat one of them and you also influence the other. This has been great news because there's a lot of biotech interest and there's lots of research of developing gerotherapeutics against some of those hallmarks with pretty much great success. And we can summarize a lot of years of research and say that healthy lifespan has been extended in numerous animal models and that relevant drugs have been used in humans. I'd like you to meet the subjects I'm going to discuss more in depth. And this is a picture of our poster child. Those are 4 children that were born between 1910 and 1920 in New York City. And the interesting thing about them, all of them passed the age of 102. In fact, it's the little sister that died at 102 when the others lived to be 110. and 107 and 109. We have 750 people like that. And another thing, they're all Ashkenazi Jews because of two reasons. Because it's a genetic study where we're trying to find the resiliency to Alzheimer and other diseases. And we need to work in a homogeneous genetically population. And a lot of the good genetic studies came from Iceland, from Amish people. When we have more homogeneous population, it's better. Also, the Ashkenazi Jews have a very homogeneous education level or socioeconomy level, and that plays a major role in health span in the United States, actually everywhere in the world. We have two studies. One is the study when we recruit centenarians and their children, and another study is where we look at children of centenarians, offspring of centenarians longitudinally. And I'll kind of mention a few things about that. Now, the first thing that we want to be sure is that our centenarians don't get sick when everybody gets sick, and now they're just sick for longer. So you see here two studies of centenarians that were harmonized. The black and red are the New England centenarian study, and the green and blue are our study. So let's talk about it. It's kind of the same. But you see that in our studies, everybody is healthy in the beginning, and after the age of 60, we start to accumulate diseases. At age 80, maybe 10% don't have a disease. But you see that centenarians in blue, they have an extended health span. They live not only longer, but 30 years longer, 20, 30 years longer than others. And even at the age of 100, 30% of them don't have any disease. Now, if you think this is cool, that's not really the important thing. The more important thing, that they have a contraction of morbidity. In other words, at the end of their life, they're sick for very small, for very short time. Those 30% of centenarians that don't have disease, they don't wake up one morning. And this is very important. And it's also confirmed by the CDC, who looked at the medical costs at the last 2 years of life of somebody who dies over the age of 100, or those who dies between 60 and 70, and it's third the cost. So there is a longevity dividend that we also underestimated, because it's one thing to have less medical costs, but if you don't have medical costs, what do? You're traveling, you're buying things for your kids, your value for the economy is really great. And we really have to get an increase of health span for the individual, but also for the economy of countries. So our hypotheses about those centenarians are, A, is it possible that they interact with the environment? And there's no doubt that the environment is important for the prevention of Alzheimer, right? Whether it's exercise or preventing obesity or certain food. Is it that 1 out of 10,000, that's the rate of centenarians in population, 1 out of 10,000 just happen to have the perfect genome? Okay, we know that there are risks for Alzheimer and for other disease, but maybe they just don't have the risk. And if that's not the case, maybe they have something that protects them. So let's look first of all at the environment. And we showed it long ago, hasn't changed much. Overweight and obesity is almost in half of our population. Smoking in 60% of the men and 30% of the women. Alcohol daily, not enough. Physical activity, even moderate physical activity, in less than half. Vegetarian, 2%. And by the way, their cohort was the enhanced 1 cohort. So they're doing either the same or a little worse than the other cohort. And the bottom line is they haven't done as a group anything special with the environment. They got there anyhow, in spite of smoking cigarettes and all that. So is it possible that they have the perfect genome? And I'll show you just one study, although you can look and see that we repeated that. But this is really dramatic, because this is when we got the whole genome sequencing of our first 44 Ashkenazi Jewish centenarian. And at that time, we had a database that's much bigger now. It's called ClinVar, and they had about 15,000 pathogenic variants that were defined. If you had this variant, you're likely to have a disease. And we didn't have a control here. We just had centenarians, and we said, well, you know, if they don't have any variant, that tells us maybe something. We were not ready to find in 44 people over 230 variants that should have made them sick. In other words, each one of them had an average of 6 variants or so, and they didn't have any one of those diseases. And you're asking, are those variants important? Absolutely. For example, we have 2 centenarians who are ApoE4 homozygote, which the textbook that Amos wrote says that they'll be demented at 70 and dead at 80, and they're 100 and not demented. So other degenerative disease and neoplastic disease and cardiac disease and other dominant and recessive variants and other things. And the bottom line is, no, they don't have a perfect genome. Okay, it really more supports the idea that they have longevity genes or protective genes or genes that slows their aging that allow them to get there. So we did a whole exome sequencing of our population, and we looked at rare variants, less than 1% of the population, and asked, are those variants expressed more in our centenarians than in unrelated control? So what Zhang Dong did, and Zhang Dong is a brilliant computational guy, actually an AI guy, geneticist. He did what we do usually, took those 1% and identified if there increase in centenarians versus unrelated Jewish control. But it didn't stop there. And the reason it didn't stop there, that we're doing something really stupid when we do genetics. We take one variant and we ask, is this variant associated with disease or not associated with disease? And we know that we're built with many variants, okay? And we can have variants upstream or downstream. So what we are doing now is we're actually networking so that all the variants go into pathways. And the way to do that is increasing in quality. And we can do a pretty job in getting those genotypes into pathways. And the bottom line is, when we get them into the pathway, the interesting thing that happened is that everything that we knew from animals for longevity was actually true here. The first pathways that came up were MAP signaling pathway, insulin IGF, growth hormone signaling pathway, and mTOR signaling pathway. So first of all, we confirmed that our animal model and our intervention have really been pretty related to humans. And also, we got many opportunities to look at specific genotypes apropos certain phenotype. And I'll give you just one example here, it's still unpublished. We found genes that were at risk for Alzheimer, and here are the first 50 candidates. And those that are in red are ones that we already knew could be related to cognition and Alzheimer. As an example, I'll give you the ABCA1, which is an interesting, really lipid gene. It interacts with ApoE, and there's a whole science around it. But how did it look in our study? We had 36 centenarians that had a SNP in this gene versus 17 of the control. So it's still small numbers, but what we can do with those small numbers when we find them is take them in the lab and work on cells. We can express those mutations in neurons or in other cells and look at the resiliency against aging, basically against against hydrogen peroxide or radiation, and kind of get the feeling if those cells are really resilient to whatever phenotype we're trying to find. So this is example for what we do with genetics. Now, let me tell you what we do with biomarkers. I should probably say something, I'm not sure that I said it before, but the mechanisms by which cell age is relatively common to all cells, okay? We're talking about something that is common, even though because of epigenetic differences, tissues are different, but it's the same mechanisms that will cause aging, or the same gerotherapeutics that will prevent aging will be relevant to several diseases. I'll give an example in a second. So we're trying to find biomarkers for aging, and as you know, it's difficult to identify early biomarkers for Alzheimer, and certainly biomarkers that will react to treatment so that you see if the treatment is working. So we need biomarkers that wouldn't only tell us what is the biological age and not the chronological age, but also biomarkers that will change when we're treated somebody with gerotherapeutics, with something that is anti-aging. So we use this new technology, SOMAmer, and I'm not going to get into details here, but we looked at 5,000 proteins in 1,000 subjects between the ages 65 and 95. That was done by Sanish Satyan, and I'm not going into the details here, but I want to show you the Volcano plot. And again, just in case people are more novice, the Volcano plot is you take all the data into this mountain that spits those red hot rocks, and the higher they go, the higher is the significant value. That's the y-axis. So this is, those two proteins are 10 to the minus 80. And the farther they go to the right, they're over, their concentration is high. And like this protein, the concentration is really fold higher as we age. And here they are low. And you see that there is a lot of biomarker, proteomic biomarkers for aging, but it is a problem. So for example, PTN and MIC1, if you overexpress them in animal, they live longer. So definitely some of those proteins are trying to prevent the effect of aging. They're protective. We don't want to take them down. Okay, and we actually want to take them up, but there's others, inflammatory markers, other that we do want to take them down, but it's hard to determine, and that's the challenge that we have. But when we do the proteomic pathway analysis, and there are many ways to do it. First of all, we get always on the top, whether it was genetics or proteomic things that are associated with insulin, like growth factor, growth hormone, IGF, insulin, they always come up first. But we've noticed that a lot of the pathways that are coming up are breakdown of extracellular metrics, degradation of extracellular metrics, platelet degranulation, collagen degradation, neutrophil degranulation, collagen biosynthesis, etc. And at first I thought, okay, just, it doesn't tell me much. Until I realized that maybe those are going to be the best biomarkers, because no matter how we're going to stop aging, we have to stop the breakdown. And maybe those proteomics are going to be really very effective biomarkers for aging. I told you there were 1,000 people. Well, 500 of them were offspring of parents with usual survival in our cohort, and 500 of them were offspring of parents with exceptional longevity. They're the children of centenarians, and our children of centenarians are basically 10 years younger than the regular people. And you can see it on the proteomic. You can see how red it is at the opus and how white it is in the opal. So in those who are with usual survival, they have 585 significant proteins that are changing with aging, and only 235 in the offspring of centenarians. Now, that doesn't mean that those proteins are changing with treatment, but what it really means that if we wait 10 years, the offspring of parents with exceptional longevity will probably have proteins that are similar to those guys. Are those related to cognition? Well, in our cohort, we don't have enough people who deteriorated cognitively or got Alzheimer. So there are 82 participants that we compared to the others, so that's not much. And we got a proteomic here, but you see that none of them cross the threshold of above 10 to the minus 5. So it's just a start, and we'll have to figure it out. When we look at the components of the cognition, such as cognition composite and visual motor, memory, attention, executive language, we kind of get similar proteins. The one that comes a lot is the MIC1, or as it's also known as GDF15. It's a very interesting one. But this is a work in progress, and I don't really have a good proteome for that predicts Alzheimer or cognitive decline. I want to end in the last few minutes with what can we do now, and how do we think of gerotherapeutics? And if you have a real gerotherapeutics that you want to try, you should recruit elderly patients to a 5-year trial and give them either the drug that you're tasting and placebo, and see that mortality is significantly decreased. So is there a drug that does it? Well, there's a drug called metformin, or in Israel, it's called glucophage. It's been used for 70 years to treat diabetes. But if you give it to animals, it extends their health span and lifespan. And there is actually a study with more than 180,000 subjects where the investigator went to the pharmacy for people who are newly diagnosed with diabetes and treated by the same doctors, and you can really look at several other factors in the health of those patients, the other medications that they take. And it was interesting that in 5-year observation, those that were on metformin had half the mortality of those that were on another drugs that's called sulfonylurea. What was even more surprising, that they have non-diabetic control, and the non-diabetic control had more mortality than the people on metformin. And so what we're trying to do is to ask, what happens if we give metformin to non-diabetic people? Now, please understand, the people on metformin have diabetes, they're more obese than the control, they have more diseases than the control, and yet with those huge numbers, they have significantly less mortality. By the way, metformin was used to prevent flu, malaria, inflammation when it was discovered that it lowers glucose, and that's how it was hijacked for diabetes. It's been used over 70 years, so it's very cheap and safe, and it's generic. And so you may be, and by the way, this is the study itself. I showed a cartoon. I want to emphasize this was not a clinical study. There is a clinical study that shows that there is less mortality on people with metformin. But this is just a unique study that, because of the amount of people they have, was really caught our attention. So why aren't we starting with metformin? It looks like the metformin decreases mortality. And the point is that usually when you have a biological discovery, you develop a drug and you test the drug, and then you need better drugs. So you're developing more drugs. It keeps the academician busy, the pharmaceuticals busy, but the FDA doesn't recognize aging as a preventable condition. Okay, so if aging is not a preventable condition, healthcare providers will not pay their clients to use a drug. And if that happens, pharmaceutical that needs business plan are not going to jump in and develop better drugs and combination drug. And that's why what we're doing, we are leading a study. I'm sorry, first I have to show you that metformin does other things. Metformin prevents diabetes in clinical study. It prevents cardiovascular disease in clinical studies. Lots of association studies, over 300 studies where people on metformin have less cancers, all cancers. Metformin delays cognitive decline and Alzheimer's. So there are clinical study, 2 clinical studies on MCI. People are taking metformin either half a year later or a year later and having some effects on some domains. There are more studies coming, and also studies, association studies with Alzheimer. And I showed you the mortality. So I'm leading a study through American Federation of Aging Research that's called Taming or Targeting Aging with Metformin, where we're going to take a bunch of elderly people, 65 to 79, 3,000 people, double-blind control study. And we're looking at the time to incidence for any major related disease, MI, stroke, cancer, CHF, MCI, dementia, or death. Let me say differently now. We don't care what disease you have, and we don't really care specifically what disease you're going to get. You know, if your mother is diabetic and you're obese, you're going to get diabetes. Because aging is what brings up the disease, you're getting 1 point over time for every disease that you're going to get. We're just going to move this cluster, okay? That's very unique about TAME. We discussed it with the FDA and we got their comments, and they liked it. This study will be helpful for many other things. For example, the NIA is paying for an omic biomarker because we want to know really what changed with therapy. As I told you before, all the studies have been done separately, but not as something that we call aging, and the FDA has accepted as, you know, if you do that, then we can approve it. So I want to conclude. So, you know, I showed you some genetics of resiliency. I told you a little bit about biomarkers and how we could use biomarkers maybe to predict Alzheimer's. I talked about proteomic biomarkers. I talked with you about metformin, a drug that also prevents decline in cognition and Alzheimer. And the future is even more exciting. You know, this is a blastocyte. You can take a sperm of a 90-year-old man and an egg of a 50-year-old woman, and you can have a normal baby. Now, what's unique about this blastocyte is that aging is reset. We don't remember the age of our parents, although we can measure the aging of the sperm and the egg. The aging in this blastocyte is 0. We figured out how to do it in this example, and we're trying to figure out, and there's some successes of how do we erase aging. And the future is you're 20 years old, you're going to get a treatment every few months or a year or something, and it's going to erase your aging, and maybe we can do better health span and lifespan then. I also want to just say that this is not only about aging, preventing aging, it's diseases. People, for example, who are going through chemotherapy are aging rapidly. People with HIV get diseases 10 years earlier. People who are disabled, I would say that poor people, there's a lot of people that need help. And so it's not only about those elderly people. Those drugs are important. Certainly, if we want to go to Mars, we're not getting to Mars without stopping aging, because on the way to Mars, we'll get cancer and heart attacks, and we'll never make it back. So we hope to together with you to stop aging, and by that to stop the cognitive decline. Thank you very much for this opportunity, and good luck for the rest of the day.